files in Dropbox. I'm glad that you remembered to turn that on. So since I just remembered to turn on the video, this is an introduction to microbiology as a discipline. And I was waiting for the computer to do updates, which took forever, so I started out doing some key concepts that define microbes. They are really numerous. They are found everywhere on Earth. They are very diverse, especially in their metabolism. What can they break down, and what can they produce as a byproduct? The metabolism part is incredibly important, and that's mostly what Unit 3 is about in this class. Microbes as a group are really ancient. You may think of them as primitive, but they do incredibly complex things, sometimes on incredibly limited resources, which is amazing. And because they are so small, they can form huge populations quickly if their conditions are good, and that has a lot of implications for human health. So I keep my files and basically my electronic life on Dropbox, so I'm going to go get my Dropbox slides now. And I'm actually going to start off with history of microbes and the history of microbiology as a discipline. Once in a while, I will have a slide set that has huge numbers of slides, but yours are broken up into separate parts so that your file size on Blackboard is more manageable. So this slide set has slides for both today and the next lecture. And as I'm going through the history, what I want you to remember about the history is generally the names of the big figures and generally what they did to advance the field. And while this is loading, so the early slides that I've gone on here have to do with size and scale and some of the big themes I already talked about. So when we talk about microbes being small, we're going to learn to do this next week in lab, where we spread out bacterial cells so that they form individual colonies. And this little spot here looks very small to us, but if we look at it under the microscope, it is jam-packed with cells. So one challenge in microbiology is to get yourself to think about life at that scale and how many individual organisms are in a tube, on a plate, on your hand, in your hair, all different kinds of places. Some of the microbes that we will talk about this semester are not actually alive. Those are the viruses. So microbe is defined by being small. Viruses are small. They are not alive. So that's something interesting to think about. We'll talk about later, why are they not alive? And some of the microbes that we will talk about are so primitive that they're like on the edge of being alive. They have barely any nutrients. They do, they live on almost nothing. They're barely alive. But they're all small, and that's what microbes have in common. I'm going to skip over these properties of life. What makes something alive or not. Because microbes are so small and numerous and diverse, and they live everywhere, they are always in some kind of community. They might be in a community with other microbes just like them. They might be in communities where they're mixed together with other microbes. They might be in communities with big organisms like us. We live in community with our own microbial flora, and that relationship is important. So we will study them, how they behave individually, and also what are their effects on their communities, and how does their environment affect them. So there is some aspect to ecology in what we talk about in this class too. Microbes are old. This is a time scale of history of Earth up until about this point. It's all microbes, and only microbes up until that point. And they've changed the history of the Earth and made Earth what it is today. We'll talk about some of that later in the semester as well. All of the types of microbes, so this is something to note down. Microbes as a group can be viral, which is not alive in the first place. They can be prokaryotic, or they can be eukaryotic. And some of the eukaryotic microbes might be certain types of protists, algae, yeast is one. Most of this class, I'm only talking about prokaryotes, not very much eukaryotes. They're still important. But within the prokaryotes, they might be bacterial in their classification, or they might be, I spelled this wrong, but I'm trying to write archaeal. They might be archaea. In this class, we'll talk about both bacteria and archaea and how they're the same and different from eukaryotes, but all of those are considered to come from one universal, last universal common ancestor. So even when we study microbes that seem completely not like us, they still have something to teach us that will relate to our own body function as well. These, I'm going to skip through a little bit. When I said that they are found everywhere, if you take all the microbes that have been found in the world at the date that this was published, 66% of them are under the ocean floor. Is that shocking? Of all the microbes in the world, it seems that most of them are marine subsurface, not in the ocean, but in the floor of the ocean and below, which is amazing. Another huge group of them are below the surface of the earth in the soil and the rocks, and so on. That's almost 3/4 of them. In what we think of as soil, about 5%, about 2% in the oceans, and every other place, only 1%. That includes in people, on surfaces, in fresh water, in toilets, garbage dumps, 1%. So the microbes that are most studied are the ones that are medically relevant, but they are such a small proportion of the microbes that are out there. So in this class we cover some of both. Also, we are a habitat. They live in community with us. So when I go through the history of microbiology, most of it is going to be focused on microbes that cause people diseases. That's why people first started studying microbes intensively, is when they realized that they could cause disease and that we could heal or treat people by affecting their microbes. A lot of research went that way. So, and it's changing now, but when I was growing up, every every bacterium, every virus, every germ. Well, that's bad for you. You better kill them. You better get rid of them because they're going to make you sick. Do we teach kids now germs make you sick? I mean, I do for my tiny sons. That's been the paradigm all this time. Germs make you sick. There are some microbes that make you sick. There are many microbes that have caused these diseases in the red, which are pathogenic, infectious disease, have killed many people over the years, and we have a lot of focus on them. But we're moving to an understanding to know that not all microbes are germs, not all microbes are bad for us. We actually need them to live in many cases. So that's something that we'll learn more about in this class as well. We also need microbes to live based on what they do out in the environment. Plants need microbes to help them fix nitrogen and to grow better. Animals need microbes to help them digest plant-based foods. This stuff is the basis of our whole food chain. Our food chain would fall apart without microbes at the base of it. This is also stuff that we'll talk about in Unit 3, about nitrogen fixation and fermentation. The cow is not digesting the grass. The cow is holding microbes that digest the grass. And again, we teach kids, cows are special. They got all these stomachs. They can eat grass. We can't eat grass. We can eat grass. We can't digest grass. We can't get any nutrition out of it. So one reason that I like to talk about microbiology is that you haven't had formal education in it before this class. And it kind of seems new, but it's also so related to your everyday life. It's fun to teach. Microbes are useful to people in helping us ferment products that maybe can't make on our own, or it's cheaper to get them to do it. For producing biofuel, for producing alcohol, and many other products that we use. Microbes are very important to all types of global cycles, including warming. So this is a picture of people studying swamp gas, and they have stuck a tube down into mud at the bottom of the swamp. And there's methane down there because there are microbes down in the mud producing it. They're releasing the methane and lighting it on fire, which is exciting. But they're probably doing that because they're studying microbial metabolism to produce methane. All of those things are important for understanding how microbes affect the world. So here's a slide for Jacinda. One of my favorite microbial facts. In lab just now, we were looking at mouth smears. And what that was is just scraping some stuff out of a person's mouth and staining it and looking at it under a slide. And another way that you could look at it is through a scanning EM to produce this kind of picture. So each tooth in your mouth has its own stable, unique population of microbes. And that changes from back to front with the micro oxygen levels. It changes from top to bottom, and it changes tooth to tooth. So like one of your teeth is almost like a planet for the microbes that live on it. And they live there and grow there, and that's their world. And maybe you brush them off every day, which is a holocaust for them. And the remnants grow back. But the ones that are back here are different than the ones that are here. It's amazing. The whole idea of scale is how small, how many is amazing in microbiology. So let's get started some on the history. These are the major figures that I want you to know. These are not the only figures in the history of microbiology. And the history of microbiology did not stop in 1920. That's ridiculous. But this was the beginning, and the giants of the field of microbiology in the golden age of probably the quickest discoveries, I would say. And as one of the sciences, microbiology is really recent. Like, it's really recent. It only started, we would consider, in 1664. People were making all kinds of discoveries before that in other fields of science, but not this. Why did it take so long?
Can't see them.
Because you can't see them. If you can't see them, how do you know that you're there? So another thing I like to think about is, people in Bible times and ancient times, they had these plagues, they had these diseases, and we take it for granted that, like, oh yeah, it's microbes. It's because you're drinking dirty water, you didn't wash your hands, you were doing this and that. No idea back then. No concept of that, of why suddenly all these people get sick. It was so much more scary then. There's no reason. Here's this plague upon us. God is displeased with us. If you know it's microbes, that changes your perspective of disease totally. Like, oh, well, we just need to have cleaner water. It's not that God is displeased with us. There becomes a reason, and there becomes something that you can do about it to treat it. So, really, before 1700, there was not a good understanding of these organisms that we call microbes. So I'm going to go through some of these figures who were big in the field. The first one is Robert Hooke, who was English, and he lived in the 1600s. And his claim to fame is that he was the first person to see cells and write it down. He saw cells, he wrote it down, he talked about it publicly, so he gets the credit for the first person to see cells. It's possible that other people saw them, but we just don't know it because they didn't write it down. So he used a very primitive lens system that kind of looks like one of those icing squeezers. Push icing out, or a syringe, to look at different things. And he happened to look at cork, and he saw the plant cell walls, which he didn't know that they were cell walls, but he saw a pattern like this of boxes, and he called them cells because they looked like monks' little tiny rooms that they stayed in, and those were called cells, so he called these cells. And then he also discovered other small and beautiful structures like fruiting bodies of fungi. Another interesting thing about him, apparently he was a huge jerk and extremely disliked by other people around him, but he was the first to see cells. The second giant of the field is Leeuwenhoek. You do not need to know how to spell Leeuwenhoek, but I really want you to always remember who he is. He was a Dutch cloth merchant in the 1600s. And he sort of had a hobby in microscopy, and he was really good at building lenses. And in his day, lenses were just, they still can be, they were spherical, small pieces of glass. So you could grind the glass into a small sphere and it would act as a lens. And the reason that he first got interested in microscopy in the first place is so that he could, this is so funny to me, so that he could determine thread counts. Because he was a cloth merchant. So probably back then, like if you had 70 thread counts per inch, like those were some supremely great cloth. And now we have like 1,200 or something ridiculous like that. He wanted to count threads in his cloth. That's how he first got started with this. He wasn't a scientist in the beginning. But he could make something, he had some kind of secret for how he made lenses. He made them way better than anybody else in his day. So here's his microscope, and there's something like, we know he made a lot, And there's something like 14 of them that exist, and we know there's others out there. And this is what they look like. Like it could show up on Antique Roadshow, and people are, I don't know what this hinge thing is. Oh my lord, it's a low note microscope. So there's the lens, and it has a screw that you screw up and down. That's like raising and lowering the stage. And he would put whatever little item that he was interested in on the tip of that needle screw thing, and he would raise it up to where the lens is. And then he would look at it. And he could see amazing detail with this. So these are his drawings. When you get to take pictures with your phone, you don't have to do drawings like this. But back in the day, you had to draw it if you were going to communicate about it. And this is what he drew. And they were accurately sized and shaped. So this is a cluster of bacteria that grow in a cluster. He saw rod-shaped bacteria, although he didn't know what they were. And what's that CD thing? What does that show? He draw the actual path that the thing took from point C to point D. It went exactly like that. And he drew it. He had like this beautiful penmanship. And he called these things that he saw animalcules. I love it. Animalcules. They moved like animals. They were tiny. He called them animalcules. And this C picture right here is someone using one of his microscopes to look at bacteria today. Amazing. They still work today. Still amazing. He's my favorite, really. And this picture, he had his picture painted when he became more famous, and that he's holding his microscope, one of them, right there. So this showed that they were small, moving, probably alive things that were smaller than we could have conceived of, that were probably alive. And that was a big breakthrough. And you couldn't know that until you had the technology, the microscope, to look at it. So since that time, microscopy has advanced a lot, but it's still using the same principles. And there's three major types. We use light microscopy in our lab. So this is coming from a light microscope. That's what we use today. And we use compound lens systems. So when we're looking through the microscope, we're using the ocular lens, where our eyes are, to magnify it along with the objective. And we're doing compound magnification. You can also use electron beams to magnify and resolve your items as well. If you see a picture like this where it looks 3D, and like you can see shadowing and so on, that's scanning electron microscope, or SEM. So this picture right here is the SEM, which is scanning electron microscope. And then the next one, where it looks more like a cross-section or a flat 2D image, that's transmission electron microscope, TRON microscope, or TEM. So if you're reading a paper or something and it says TEM 10,000x, transmission electron microscopy 10,000x magnification. And sometimes these will be false colored, but they're always in grayscale, the actual data that comes out. What I want to do right now is I'm going to switch over to the microscopy slide set. And if you don't have that printed out, it's okay. It's not complicated information, but since we started doing microscopy today, I'm going to go ahead and talk about that information. And I will post this slide set on Blackboard tomorrow after lecture. not there yet. And I'll actually stop after microscopy and pick up with more history tomorrow. But since Lowenhoek pretty much developed the first microscope that was high quality, we'll go ahead and do microscopy here. So microscopy can even be something as simple as using a magnifying glass with one lens to magnify objects. How many of you are in Physics 2 this semester? Yeah, you probably will be doing like basic light bending and lenses and so on and why it magnifies. I'm not getting into that here. But if you're using lenses to magnify objects, that's a type of microscopy. And the lenses are bending light. This is the picture you need for your studying your microscope parts that I talked about. And this is a compound microscope. It's got ocular and objective lenses. What they call the substage condenser, I'm calling iris diaphragm lever, is what I call it in our lab. I'll take either terminology. Oh, they are. They're showing it right here. Aperture diaphragm control, same thing as the iris diaphragm lever. And this microscope is a little bit different down at the bottom than ours is. So their blue filter does not float like ours, but the other parts are the same. So when we use lenses to make images bigger, that's magnification. And we definitely need to be able to magnify microbes to make them appear bigger so that we can see them. But if we just make the image bigger and we don't also make it resolved or sharper, we won't really be able to see very much. So one thing I want you to know after this lecture is what's the difference between magnifying something versus resolving it, or what's the difference between magnification and resolution. If you just make something bigger, that's magnification. If you make it more defined or crisp, so to speak, that's resolution. So when Han and I first got married, I think we took a tax refund or something like that. It was a big amount of money, and we bought a digital camera. And it was so expensive, and it was 2 megapixel. Lame now. 2 megapixel. And we take all these pictures. And then if we got one that was really good, you're like, oh, let's blow this up and put it on the wall. If you take a 2 megapixel picture and you blow it up, it gets fuzzy. So yeah, it's bigger, but it's grainy, it's fuzzy. You magnified, but you didn't resolve it. Magnify that. That'd be scary. It's a stink bug. If you want it to still look good, it also has to have resolution. So if we just magnify and we don't resolve, we're not going to get useful information out of our image. What these balls, spheres here are showing, pretend I'm writing this on the slide, those are related to the wavelength of light. So this is showing what if we're illuminating this with light that has a big wavelength, versus light that has a small wavelength. And if the light has a small wavelength, you can think of it as it gets into the nooks and crannies better around the object. Does that kind of make sense? If you really want to resolve an object, you need to use short wavelength illumination. So write this on here. This side is shorter wavelength. That's a symbol for wavelength illumination. This side is longer wavelength illumination. Which type gives you better resolution? Shorter wavelength. I think of it as it fits into the nooks and crannies better. There's another picture that I need. It is not here, so I'm going to draw it over here. So if we think about the spectrum of electromagnetic energy, at one end we have like gamma rays, X-rays, high intensity, really damaging stuff. At the other end we have infrared and microwaves. And in the middle, we have visible light in the middle of the spectrum. And as you go toward this end is shorter wavelength and higher energy. That end is the opposite. You can fill that in later. So for really good resolution, if we really want to get light or other electromagnetic energy into the nooks and crannies around small objects, we need to be toward this end. Visible light is in the middle. We use visible light in the microscopes down the hall. And visible light, the colors of it are arranged this way. I learned it in school as Roy G. Biv, and I've got it drawn backwards this way. But visible light, the best that we can do with visible light to get short wavelength is to get on the blue end of the spectrum, and that's why you've got your blue filter. I should have had you take off your blue filter and look at the image and see how much more blurry it is. We'll do that next time. So to get good resolution, you need short wavelength light. The electron microscopes, I don't have that slide on here. Electron microscopes are down in this range. They can have, those electron beams can have shorter wavelength than visible light, so you can look at smaller objects with them. That's why. Another thing that we can do to kind of push the limits of our light microscopes, those light microscopes, like we're pushing the limits of how small can we see with those. We use short wavelength lights, we use compound lenses, ocular and objective, and we also use oil between the lens and the slide to trap the light and keep it from scattering. That's why the oil is there. It keeps more light in the system, and that helps improve the resolution. Again, if you use the oil lens without oil, you won't get good resolution. It'll look more fuzzy. You can do it, it just doesn't have as good of a resolution. This slide you probably can skip over, but do you know how to calculate the total magnification when you use compound lenses? You may have learned this like in a gen bio or high school and forgot, but I want you to know this too. If we're using two different lenses, and each one has its own magnifying power, we can use that information to calculate the total magnification. Lens power times lens power. And for our microscopes, the first lens is the ocular, and it's always 10 mils. That doesn't change. The other lens that we're using is the objective. And for our microscopes, that's either going to be 4x, which means if I just look through that, the thing looks 4 times bigger. It'll be 4, 10, 40, or 1000x. So what is the highest magnification? Getting ahead of myself, 100x. What is the highest magnification that we can get out of our microscopes down the hall? What'd you say? It's 1,000. Yeah. 1,000x is the best that we can do with the light microscopes that we have. If it needs to be bigger than that, you really need to go to an electron microscope. So what can we see with our microscopes? Or what can we magnify and resolve to a usable level? We can get down with our microscopes, that are light microscopes, to maybe 0.2 microns. Micron is a millionth of a meter. 1,000th of a millimeter. Whoa. 1,000th of a millimeter. We can use my light microscope to see that. It's great. I love it. So small. If it's smaller than that, we will need a different type of microscope. I wanted to skip a little bit ahead. No, I think I did. I think I'm going to stop there. There's some other parts in this slide set about staining, and I think I'll go through that when we actually do staining in lab. So, for tomorrow, you'll need to read the section about history and also some microbial diversity. We'll have that quiz tomorrow. 8 a.m. across the hall.